Welcome back my course aspects of biochemical engineering. So, last two lectures I tried to discuss about the stoichiometry of bioprocess. Now, this will be I will be completing this stoichiometric bioprocess by this lecture, and here I shall discuss two other problems, and I hope that this will clear all the that whatever problem that we will be having in the stoichiometry. Then I request all of you to do this stoichiometry of this equation by as your own, so that you practice it, and if you have any problem, please let me know. Now, first problem that I am going to discuss in this lecture, this is the anaerobic digestion process. As you know, the anaerobic digestion process is largely in operation in India and throughout the world, particularly anaerobic digestion process, because if you look the history of the wastewater treatment processes like this, that initially all the wastewater treatment processes we considered kind of wastage for the in the industry. And why we have this wastewater treatment processes? The reason is that no industry, particularly chemical and biochemical industry, they cannot discharge their wastewater directly to the water courses, because as soon as they discharge this to the water courses, that water will be polluted. A lot of bacteria will grow in the water and pollute our drinking water sources, and it is a serious concern. So, we shall have to treat the organic waste. Now, it has been observed for all the industry that 10% of the total investment they keep for the wastewater treatment processes. Now, initially I told you that 70% of this wastewater treatment processes are controlled by the biological means, and 30% maybe through chemicals and other means. So, now this process, when you go for the wastewater treatment process, initially we thought that it is a kind of wastage of money. Now, due to the development of the anaerobic digestion process, it is now possible that a part of the money that you spend for wastewater treatment process can be recovered. I can give a typical example that in West Bengal we have IFB agro industry. They produce the ethanol from broken rice, and they have the anaerobic the treatment process, and they told me the 50% of their distillation cost, distillation energy recovery, is taken care by the anaerobic digestion process, that is methane that is produced by this process. Now, the problem that I am going to discuss now, this is the anaerobic digestion process, and this now problem is like this, that elemental analysis of refuse contains 76% weight by weight, that is the of organic matter. Actually, you might be aware that organic matter only participate in the biochemical reaction mostly, and inorganic matter also can participate, that is used as a cofactor for different enzymatic reactions. That is the only thing that we have. Now, this empirical formula of the refuse is given here, and remaining 24% is the ash. Ash is kind of metal oxide, and the minimum methane production through the anaerobic digestion is to be calculated per kg of refuse, and also determine the stoichiometry of the process. So, that I hope the idea is very clear, that some organic waste is there, we putting it in the anaerobic digestion process, and we want to produce the methane. So, we shall have to find out that what is how much methane we can produce out of that, and not only that, we shall have to also find out what is the stoichiometry of this process. Now, for doing so, question comes, what is the basic equation of this? So, basic equation will be this. This is the substrate that we know, that is already given here, that is C H2.1, O.9 and N 0.15. This is this plus water molecule, this is not given here, maybe this should be given here. This is This because I told you that waste material comprises of that lot of polymeric material like starch, glucose, kind of protein. So this undergo hydrolysis. Hydrolysis means the reaction which is take place in presence of water. When a hydrolysis, then it forms the smaller molecules, and these smaller molecules then participate in the reaction and produce methane, carbon dioxide, ammonia, and H ion that is produced. So we shall have to develop the stoichiometric equation on the basis of that. So that is the whole thing, that when you do the analysis of anaerobic digestion process, the preliminary concept on the process is very much required. Until unless we have the conception of the process, we cannot write the stoichiometry of the equation. Conception, I mean that what are the initial requirement of the process, what is the final desired product we are going to get. If you have some idea, then and only then you can write the tentative equation, and then whatever coefficient is there, we can fill it up one after another. I can show you. Now this is the tentative the reaction that we have in the anaerobic digestion process, and as you know that anaerobic digestion process is usually carried out by two group of microflora. One is acidogens and methanogens. Acidogens means that convert the organic matter to the organic acid, volatile fatty acid, and volatile fatty acid will be converted to the to methane and carbon dioxide. Now, this is the ultimate equation that we have here. Now, here we can find out sigma s and gamma s we can find out, and sigma p and gamma p we can easily calculate on the basis of this that the empirical relationship we can find out that. Now, this anaerobic digestion process, what we have, we know this the thermodynamic coefficient is 0.7. I have already mentioned. So, if we, and here in this stoichiometric equation, we have seen that in this stoichiometric equation we have seen that in the stoichiometric equation we have seen only product. This product we have not considered. This is the product we have considered, but we didn't consider any cell mass here. Cell mass, because the anaerobic fermentation process, cell mass production we can appears to be negligible. It is 1/10 as that of aerobic fermentation process. So this we neglected. Now, so y p by s is equal to epsilon p sigma s gamma s sigma p gamma p. So in this all coefficient is given here, and so if we solve this equation, it is coming about 0.78 gram of methane produced from gram of substrate. So this way we can find it out. Now here the gram of substrate, when you consider, this is the organic substrate, but this again comprises of 76%. We have seen the refuse contains 76% organic matter. So, if you consider actual refuse, actual biomass, then what you have to do, we shall have to multiply it by 0.76, because 76% is the organic matter present in the biomass. So, if you multiply 0.6, then we find out gram of refuse, that this much of gram of methane produced per gram of refuse. Now, previously it was per gram of methane produced per gram of substrate. Now, we can calculate it the per gram of refuse. So, this is coming this has come at 0.134 gram methane per gram of refuse. And how you can calculate the stoichiometry of this process? We can do it very easily. That if we know this, we can find out. We know the what is the molecular weight of methane and what is the molecular weight of biomass. So from that we can find out. If you look at our coefficient, this we have found out. We have found out how much B is produced. We can easily calculate B value. We can easily calculate. If you know how much B value is produced, then we can easily calculate the carbon dioxide, because if you do the carbon balance, 1 equal to B into B plus C. Am I right? So if you know we know this is 1 and this is B value you calculated, so you can easily calculate the value of C. So this is how you can find out the we can calculate the value of C here. Value of C you can calculate. This is coming like this. Then we do the nitrogen balance, oxygen balance, hydrogen balance. We go as I mentioned in the previous lecture, that we simple the kind of the balance equation, that if we do that, we can easily solve this equation. That this is like this is coming like this. You can solve all this all this coefficient. We can easily calculate. Now, when you have all the coefficient, then we can put this equation that we have already written previously. This equation will come. So this will give you the total idea that what is the stoichiometry of this process. Now here I want to add something else. Suppose this is the molecular conversion. Am I right? This is like this. If you like 0.33 gram atom methane per gram atom refuse. Am I right? Now we know that what is the atomic weight of this, and we know what is the molecular weight of this. This molecular weight and this molecular weight. So we multiply by that. We can multiply. Then we will get the gram of methane produced per gram of substrate. From that you can do that. Now now let me show you something else here. Now suppose you guess this much of methane produced. So you can find out 0.33 moles of methane. Am I right? So now we know at NTP. Into what is NTP? NTP is normal temperature and pressure. What is the normal temperature is pressure? Temperature is 0 degree centigrade and pressure is 1 atmospheric pressure. That at NTP 1 mole of any gas, this is what is the volume that we have? 22.4 liter. Am I right? So if you know this much of moles, so if you multiply it by 22.4 into 0.33, then this liter of methane will be produced per gram atom of this refuse. So now if you divide by molecular weight of this refuse, then you can find out the per gram of refuse how much methane is produced. That you can easily calculate. Okay. Now let me go to the next problem. Next problem is very interesting. If you look at here, the microorganism Microbacterium vaccae is able to grow with ethane as a sole source of carbon and energy. Now here I want to point out one thing, that that I discussed that single cell protein may be considered as a uh, very good source for the animal feed, and it is not very good for the human food. The reason is that it contains lot of nucleic acid, and due to presence of nucleic acid it causes gout and kidney stones. So, there is a trend in the world that how we can produce the microbial proteins. So, different countries they are producing microbial protein from the different sources. Now, in our country we have very less petroleum reserve. We have hardly, we have whatever petroleum reserve we have, mostly it is be consumed and only for 30 years, not more. That is why most of the crude petroleum we import from outside. Now, in the petroleum producing country, they have lot of petroleum. They can use the hydrocarbons for the production of cell mass. So, this is the problem related to that. Now, they use the ethane as a source of carbon. Ethane is kind of hydrocarbons, that this is used as a, for the production of mycobacterium. It grows at 30 degrees centigrade in a CSTR chemostat with a continuous supply of gas, 5% ethane and 95% air, and of a sterile aqueous media which contain various minerals, nitrogen as ammonium sulfate, and limited the substrate in is ethane. What is mean by limited substrate? Limited substrate, which is the what is the component that present in the limited amount? As since ethane is a hydrocarbon, is sparingly soluble in water, and you know that microorganism can utilize the substrate which is soluble in water. They cannot take the substrate which is insoluble. So, it's first it is to be solubilized. The solubility, since the solubility of ethane is very less, so that is why the ethane solubility is a limiting factor in this particular reactions. And yield coefficient, this is very important. Yield coefficient is 23.7 gram cell per mole of ethane. That is given there. Now, except for the small amount sulfur and phosphorus, an analysis of dry mass carbon content is 47.4% weight percent. Weight percent means weight by weight. That means if you take the 100 gram, basis is 100 gram, then carbon content in the biomass is 47.4 gram. Then nitrogen 8.3 gram, hydrogen is 7.43 gram, as is 4 gram. The remainder is taken as the oxygen, which cannot be detected in the analysis. It is very difficult to find out oxygen present in the substrate. Determination of the stoichiometric formula of the ash-free biomass is A, H, A, O, B and N, C, and the formula weight per gram atom carbon, that and determined Y x by S in the unit carbon mole biomass per carbon mole ethane. Now you can remember that previously what is given? This in this problem that is given, this is like this. The gram of cell mass, 23.7 gram of cell mass produced per gram of ethane. So, this you have to calculate the gram atom of cell mass produced per gram atom of ethane. That we shall have to find it out. This is the equation that we have, gram mole biomass per gram. This is, then again we shall have to calculate the oxygen consumption, that gram molecular oxygen per gram mole of biomass formation, and when it is assumed that carbon dioxide is the only product besides the biomass, and write the full stoichiometric equation and determine the total heat evolved per kg of dry cell mass formation. So, this is very interesting in the sense that here you get the opportunity to find out how to determine the empirical formula of the biomass, and then you have to calculate that carbon mole biomass produced per carbon mole ethane consumed. Then you calculate what is the oxygen consumed, then followed by the stoichiometry of the whole process, and finally you have to find out the how much heat evolved in this process. Now, so how you are going to solve this? This is very interesting. That suppose that that in this that if you look at the biomass, so we have carbon. Am I right? Carbon is 47%, 37.4. This is basis is 100 gram. Basis is 100 gram biomass. If 100 gram biomass, carbon present is 47, nitrogen is 8.3. This is all gram. And then we have hydrogen, this is 7.43 gram, and as is about 4 grams. Now, remaining whatever after summation all these things, whatever remaining, that will be oxygen. Remaining is oxygen. So, what I want to point to, and it contains about 4% as. So, when you do the stoichiometric analysis, we do the stoichiometric analysis with respect to the organic substrate. So, naturally that this is to be converted to the 100 gram of not biomass, 100 gram of organic material. So, if you want to convert to 100 gram of organic material, then with that carbon content it will divide by we can 0.96. That 4% is there, that you convert 100% of biomass. So, this is also 0.96, this is also 0.96, and so we can do that, this also 0.96. Now, when you get this carbon, you know that what is the atomic order of carbon is 12, nitrogen is how much, 14, hydrogen is 1, oxygen is how much, 16. So, you know that. So, if you can do the analysis carbon, hydrogen, oxygen, we can find. Then, so we shall have to find out the biomass empirical formula on the basis of 1 gram atom carbon. Am I right? 1 gram atom carbon. So, how much carbon atom is there? This is divided by 12. Now, this you have to divide with this, all these things you divide, then you can find out per gram atom of carbon how much nitrogen is there, how much hydrogen is there, how much oxygen is there. Now, if you have this, then you can put it in the equation like this. This is C H O N. So, we can write different figures as per. This is 1, but this will be different. This we can write it here. So, we can have the empirical formula like this, and then we can find out the molecular weight of this particular formula. So, this is exactly what it has done here. This is what exactly we have done it here. This is the figure that we have, and then we have find out like this. This has come like this. Now, we got the formula like this. This is the formula we got, and with the molecular weight this. Now, we have only this figure 23.7. If you divide by 24 point this molecular weight, then we will get the gram mole biomass per gram mole of ethane. Now, since it contains about 4% of as, so you multiply it by this to get the exact figure. What should be the figure of gram mole of biomass per gram mole of ethane, that you can easily find it out. Now, next question is that how we can find out the oxygen requirement of the system. So, again in this system, first we shall have to find out that what is the tentative stoichiometric equation. So, this is the ethane, this is gram atom ethane. This is usually the formula is ethane formula is C2H6. The gram atom will be CH3, and then ammonia is required here, cell mass is produced, and this is oxygen is required. This is the aerobic process. This is the cell mass, and this is carbon dioxide, and this is water. This is the what we have the equation, and from that we can find out different coefficient. This is substrate, this is the cell mass. That way we can develop the gamma S value. See, gamma B value you can calculate, and from that you can also determine this B value, because YC value already you calculated the gram moles of the biomass produced per gram of ethane form. So, if you know that, you can put this equation here, and this is coming about 1.236 that is moles of oxygen per gram atom of ethane. Now, then we can also calculate the YOC, that is this I can show you. OX means gram of oxygen, required gram of cell mass. Am I right? So, I can write like this. This is equal to Y O by S and Y X by S. So, we can easily do that, because if this SS will cancel. So, this is exactly what is shown here. What is shown here. So, you can do that, and then if you do the balance, carbon balance, and nitrogen balance, oxygen balance. Oxygen already we have calculated. We find this stoichiometric equation. This stoichiometric equation you can calculate. Then we can heat evolve. We can find out like this: 4 Q. This is 4Q0 into B. B already we calculated, so we can find out this is kilojoule per mole of substrate consumed. And then I told you, if you divide with the molecular weight, you find out that how much per gram of cell mass, how per gram of substrate, how can it produce. Then you divide by the YX by S. If you divide by YX by S, the gram of cell produced per gram of substrate consumed, then you find out for per gram of cell mass how much heat is evolved. So, this is how you can calculate that total heat evolved in this process. And so, in conclusion, I want to tell you that in the stoichiometry of the biochemical process, it can be done very easily. First, we shall have to know the different empirical formulas of the substrate. We should know the empirical formula of the biomass. We should know the empirical formula of the product, and the empirical formula can be easily defined. We can the carbon, hydrogen, oxygen, nitrogen. We can assume that it is present there, and then we can, through the empirical analysis, we can easily find out, if you know the elemental composition of this material, we can easily find out the empirical formula. Once you know this empirical formula, then we can write the different coefficient that is we required in this particular equation. And when you write any kind of biochemical equation, we should have some preliminary idea on the biochemical process, whether we are targeting for the cell mass formation, whether we are targeting for the product formation, whether the process is aerobic or anaerobic. All this information, if it is there, then on the basis of that we can write the stoichiometric equation. And also I showed you that how you can find out the validity of the experiment results, and also amount of heat evolved in the process. I hope this will be very much useful for do the mass and energy analysis of any kind of biochemical process. Thank you very much.